Good morning, beautiful people. Here I am at the beginning of a very long day for me, so I'm here a little bit earlier than usual doing this. Hopefully that'll benefit you. And here we are starting one of my favorite chapters. I hope you enjoy this topic because I know I do. As a biochemist, I want to know how life works, how molecules look, and how they work. Okay, those are the two basic questions that a biochemist or a molecular biologist would have, and that's really what drives me. Okay, and in order to answer those two questions, you need a set of tools, and that set of tools are vastly encompassed in this chapter. Okay, so this chapter is on recombinant DNA technology, and first I want to ask you if you know what recombinant DNA technology is. Okay, the word is to recombine. Okay, so what this means is that prior to the inception of recombinant DNA technology, we did not have the tools to recombine genes. Okay, we could choose animals, or we can choose plants to mate and consequently make an offspring with certain traits. And it was kind of a hit and miss thing because, as you well know, in meiosis, what genes you get from mom and what genes you get from dad, independent assortment tells you that there's no real good chance that you're going to get all the traits that you're looking for. Okay, but with recombinant technology, that is not the case, because you purposely take the gene of interest, the gene that you are interested in, okay, and you cut it out and you splice it into, these days, into a plasmid, into a plasmid is supposed to be a naturally occurring piece of circular DNA, but that's really not true anymore because even plasmids are engineered these days. Okay, so you take the gene of interest and you put it into a plasmid, and then you take that plasmid and you put it into a bacteria in order to produce the protein of that gene. Okay, there's a whole purpose behind this, is to make a particular protein that we're interested in. So the recombine part has to do with the fact that we can cut a gene very specifically with chemical scissors called restriction enzymes, and we can recombine that into a vector such as a plasmid. That's recombinant DNA, having the ability to choose a gene of interest and putting it where we want. Okay, so why would we want to do that? Well, because according to our little PowerPoint here, we're trying to get rid of traits that we don't want. We're trying to bring together those traits that we are interested in. Okay, so combine beneficial traits. And the interesting thing is that recombinant DNA allows us to grab genes from wherever we want. So the gene that we're interested in might be in a bacteria, it might be in a fungi, it might be in a plant, it might be in a human. It really doesn't matter. And we can choose those genes and put them and recombine them into a new host genome and make the protein of interest. Okay. A lot of people get worried when we talk about this. All I'm going to tell you is that recombinant DNA is a tool. Okay, it's amoral. The person that's using it is the one that chooses whether it's going to be used for a better good, such as in producing medicine, or is going to be used to undermine what we need as a species. Okay, so here's the author saying exactly the same thing that I just said in words. Okay, so you take a bacterial cell. Don't be surprised. It's probably going to be E. coli. And then here I'm going to try to enlarge it. Ooh, that enlarged nicely. That's beautiful. There's our naturally occurring piece of circular DNA. And again, that's a little bit of a lie because all plasmids these days are engineered by biochemists. Okay, you can buy these, you can trade them. They're like baseball cards. You can actually trade plasmids if you want. It's quite interesting. So here's our plasmid, okay, and here's our gene of interest, okay. And then what we're going to do is we're going to cut that gene of interest into small pieces, hopefully pieces that produce an entire protein that we are interested in studying later on. And then, again, how do we cut it? We use these beautiful things called restriction enzymes. And those restriction enzymes, we're going to cover that in just a second. Cut the gene in recognition sites. And then we have pieces of that gene. The plasmid is cut with the same restriction enzymes. And that's important because that means that the ends of this gene of interest are going to be compatible with the sticky ends of this plasmid, okay? And then you add ligase to make sure that this becomes a complete piece of DNA, so that the phosphodiester bond is created both here and here. And then you transform the bacteria by putting the plasmid back into the bacteria. And then you can save the bacteria indefinitely. You can put it in a tube like I do, or you can even lyophilize it. You extract that moisture out of it, and it can keep indefinitely. So what does this mean? What this means is that you have effectively saved that gene that you're interested in studying for as long as you want. At a later time, you can harvest it, and you can put that gene into, like it says here, into plants, or you can sequence a gene, or you can produce a protein in order to sell it in the market, or whatever. I mean, the purposes are a lot more than the 3 or 4 that are outlined here by your author. They're innumerable, okay? So one of the things that's very interesting about biotechnology, okay, says the discovery of retroviruses, okay? So let me take an aside here because this is important, okay? Since the discovery of retroviruses, HIV, human immunodeficiency virus, is a retrovirus. What does it mean? What it means is that the genetic code in that virus is not DNA, it's RNA, okay? And then it infects a host, okay, typically animal or human. And the first thing that virus has to do is it has to turn that RNA into DNA, okay? Now, if you remember the central dogma from last week, you know that you go DNA, messenger RNA, and protein. That's the flow, right? So to acknowledge that viruses can go from messenger RNA back to DNA, that's going backwards on the central dogma. And that's a discovery that we had with retroviruses, that, wow, we can go both ways. Now, you might think to yourself, who cares? We care, okay? And the reason that we care is because the majority, I'm going to say probably half of the medications that we take these days are harvested by putting a gene of interest into a bacteria, okay? But that gene of interest cannot be taken from the human DNA for a reason that I'm going to explain in just a second, okay? It has to be taken from human messenger RNA. And in order to accomplish this task, we need to take a protein, an enzyme, a machine from these retroviruses, and the name of that machine is called reverse transcriptase. What does it do? Again, that reverse transcriptase goes backwards on the central dogma. It goes from messenger RNA back to DNA. It's a beautiful thing. This thing literally opened the doors of biotechnology. That's how important this is, okay? So let's take a look at why this is important, okay? We're going to go from microorganisms to animals such as ourselves. So let's say for the sake of argument that we're making insulin, okay? And the reason I like to pick on insulin is twofold. these days you can't throw a rock without hitting somebody who's diabetic. And the second reason is because this is probably the first protein that was synthesized using this new knowledge, these new tools. And in particular, the tool I'm talking about again is the protein or the enzyme called reverse transcriptase that we harvested from retroviruses, okay? So why do we need them? Okay, so this is eukaryotic processing, okay? This is not us. Forgive me, I just said that incorrectly. That's us. This is not bacteria. This is us, animals, plants, cells that have a nucleus, okay? What happens is that we have a gene, okay? Here's our gene, here's our DNA. Do you guys see my DNA there? We have the DNA, but we are incredibly complex animals. We're very complex, okay? And if you were to look at the Human Genome Project, you would find out that we only have 30,000 genes in our whole genome. And you have to conceptualize in your mind, how is it possible that our body makes so many different proteins, yet we only have 30,000 genes? I mean, we must make easily billions or trillions of proteins, easily, and we only have 30,000 genes. How is that possible? Well, the answer is that each gene that we have can produce more than one protein, okay? And this event is called alternative splicing. So depending on what protein your body makes, your gene expresses different parts and makes different proteins. So each gene can make 3, 5. There's some genes out there that can make close to 200 different proteins, which is just incredible, okay? So here we are. Here's our DNA, all right? And notice that the DNA has these parts called introns. Here's intron 1, intron 2, intron 3. And see how the author says these are non-coding regions. In other words, these introns do not make protein. Only the parts in between, the exons, make protein, okay? And again, the reason that this is happening is because this allows nature to associate these different exons in different patterns and manufacture different proteins, okay? Well, as we come back to insulin, as it turns out, insulin is made out of two exons, not just one, but two, okay? So the reason that this is important is because if you were to take the gene for insulin and put it into a plasmid and then put it into a bacteria, the protein that you would make would be garbage. It would be meaningless. Why? Because that gene still has these non-coding introns, and those things have to be cut out, okay? So what happens in our body? In our body, we go from DNA, we go through transcription. Here's our central dogma again. We go through transcription. We make our messenger RNA, okay? This messenger RNA goes into the cytosol in order to attach itself to either the rough endoplasmic reticulum or to a free-flowing ribosome in order to carry translation, okay? But before it does that, in the lumen of the rough endoplasmic reticulum, there's a beautiful protein, a beautiful machine called the spliceosome. And what that spliceosome does is it splices out these non-coding introns out, it gets rid of them, and splices together the exons. So notice here that once this spliceosome is done, you have exon 1, 2, 3, and 4 attached here. Those are the actual coding parts of the messenger RNA that will make the correct protein once those introns have been excised, okay? So here's our messenger RNA as a 5' guanine here that protects it from being hydrolyzed and broken down into cytosol. It has a polyadenosine tail. This polyadenosine tail is susceptible to nucleic acid hydrolyzing enzymes. In other words, when this is in the cytosol, there's enzymes that are going to cut this down like a little Pac-Man and it's going to eat it up and destroy that messenger RNA. We want that to happen, okay? Because when we express a protein, we don't want that protein to be expressed forever. It has to be expressed for a certain amount of time. You make enough protein and then you shut it down. So the way that gets shut down is that, again, we have hydrolyzing enzymes that cuts this down, and then once it reaches the mRNA, no more protein production. So effectively, this polyadenosine tail here is a little timer. It's a fuse. And the more protein that the body needs to make, the longer that this poly A tail is, the less protein that needs to make, the shorter that poly A tail is. So this is a little egg timer here. And once this fuse gets eaten up and the enzyme goes into the gene itself, we don't make any more protein, okay? So all this to say that from this point here, where we have the raw messenger RNA, to this point here where we have the finished product of the messenger RNA, this event right here is called post-transcriptional processing, okay? And that post-transcriptional, because it happens after transcription, that post-transcriptional processing takes place typically in the lumen of the rough endoplasmic reticulum by the spliceosome. And the finished product is this messenger RNA that has the correct parts of the gene that need to be expressed into the protein of interest, the 5' guanine that protects in the cytosol, and this little egg timer, this little fuse down here at the end. Okay? So all that to say that if we're going to make insulin, we cannot use DNA. We also cannot use this pre-processed messenger RNA. We have to wait until the cell produces, I can't seem to move my slide, until the cell produces this post-transcriptionally modified messenger RNA, okay? And then we put chemicals in there that arrests this step so that nothing happens after this. And so we grab a cell that is rich in this messenger RNA. The problem is that we have messenger RNA. And if we're going to use recombinant DNA, it's recombinant DNA. It's not recombinant RNA. So what this means is that we have to take this proper messenger RNA and turn it back into DNA and then be able to express the protein of interest, okay? So there's our messenger RNA. Now what? Well, now we can take this messenger RNA, apply this reverse transcriptase, and if you remember, okay, RNA is single-stranded. It's not double-stranded like DNA, it's single-stranded. So this reverse transcriptase will make a complementary strand of DNA in a 5' to 3' direction over here. And then you end up with a different abomination because now you have two strands, but one is DNA and the other one is RNA. Fortunately, DNA polymerase 3 can now recognize this and substitute that last strand of RNA for DNA. And voila, now we have the DNA of the gene that we need to make in the laboratory. Fantastic. Again, all this is only doable because we have this enzyme from retroviruses called reverse transcriptase. Okay, beautiful stuff. So to recap in 3 seconds, we have DNA. That DNA undergoes transcription by RNA polymerase into the messenger RNA. That messenger RNA undergoes post-transcriptional modification via the spliceosome in order to have the post-transcriptionally modified final product of the messenger RNA down here. Then reverse transcriptase synthesizes a complementary strand of DNA on this single-stranded RNA, and because the DNA is complementary to this RNA, it is called cDNA, for complementary DNA. That means that you have a hybrid of RNA and DNA. Now DNA polymerase comes in and synthesizes the second strand of DNA, removing the RNA, okay? And then you end up with double-stranded DNA, and finally that DNA can be incorporated into a plasmid, put into bacteria, and now we can produce insulin and people can live longer, okay? All right, now we're moving on. We're gonna introduce a couple things that, some housekeeping things that are essential for you to know. And I'll just introduce it as tools, and then later on I'll elaborate on the application, okay? So the first one that we're gonna start with is the production of synthetic nucleic acids, okay? So one of the things that we make in recombinant technology is we make DNA and we make RNA, okay? In the cell produces nucleotide triphosphates. Hopefully you remember this. So it makes GTP, CTP, ATP, TTP, and it makes the same 4 nucleotides for RNA. So GTP, CTP, UTP, uracil, and ATP, adenosine triphosphate. And these are essential because we need these nucleotide triphosphates anytime that we're sequencing a small piece of DNA. And the roles of these DNAs are vast. We use them for very many different reasons. So for example, one of them is to produce the genes necessary to produce express certain proteins, okay? We're gonna talk about making DNA and RNA probes. What's a DNA or RNA probe? Well, let's say that you have a piece of DNA. You're looking for a particular sequence of nucleotides in that DNA. What you would do is you would make a complementary piece of DNA or RNA, depending on what you're using, DNA or RNA. And if it's complementary, the hydrogen bonds, the sticky ends, will be complementary to each other. So that complementary piece of DNA or RNA will only adhere to that particular nucleotide sequence in the DNA. Then what you do is you put a reporter protein in it. What's a reporter protein? A reporter protein, as the name implies, is a protein that reports to you that it's there. So what are we talking about? Well, it used to be radioactive isotopes. Thank God we've moved away from those. And now the reporter protein is usually a green fluorescent protein of some sort. So you make a complementary piece of DNA or RNA, you stick a green fluorescent protein on there, you wash your DNA with the probe. The probe will only adhere to the complementary nucleotide sequence. And then you put UV light on top of it. And if it fluoresces, you go, aha, the probe is there. So the gene of interest is there, or the sequence of interest is there, okay? So that's what a probe is, okay? A probe for DNA and RNA is a complementary piece of, is a complementary piece of DNA or RNA to a sequence of interest that has a reporter green fluorescent protein attached to it, okay? You can make antisense nucleic acids, such as interference RNA. We've been playing with those lately in order to use them as means of therapy. So effectively, if you have DNA, well, not DNA, but if you have RNA, because it's single-stranded, and you make a complementary piece of RNA, that RNA is going to stick to it, just like the probe is going to stick to a complementary region. So then when a ribosome comes along and is trying to translate that piece of mRNA, is not successful. because there's something in the tracks impeding the process of that ribosome. And that thing in the tracks is that complementary piece of interference RNA. So this is a hot topic right now. A lot of people are trying it. To be honest, we're having a lot of trouble with it because that interference RNA has to go into the cell. And I got to tell the cell does not like having nucleic acid in the cytosol. So as soon as it goes into the cell starts degrading it. But we're still playing with it. So antisense nucleic acids. And then my favorite one is the fact that we use these nucleotide triphosphates for what's called polymerase chain reaction. This is important. So I can go into PCR into more detail, but the point of it is that you cannot watch a CSI show right now without knowing about PCR, polymerase chain reaction, okay? Once upon a time, if there's a murder and somebody left a small speck of blood, they were gonna get away with a murder. That is no longer the case, because now if that speck of blood has a few strands of DNA in it, we can use polymerase chain reaction to take that DNA and multiply it to an abundance and then do an assessment of that DNA and find out who it belongs to. So this is very useful, okay? So these are the roles. This is some of the purposes for synthetic nucleotide triphosphates, all right? Moving on, we have restriction enzymes, okay? Like I said before, typically you have a gene of interest, and that gene of interest has to be incorporated into a plasmid, and then that plasmid goes into a bacteria. So what that means is that we have to cut the gene of interest and the plasmid with chemical scissors. And not only do we need to cut them with chemical scissors, we have to cut them in such a way that the ends are typically complementary, okay? And the reason that's important is because if the ends are complementary, then the complementary hydrogen bonding will cause the ends to attach to each other. And now that that gene of interest attaches itself to the plasmid by itself. You don't have to do anything, okay? So these restriction enzymes are again, they're enzymes that bacteria use to defend themselves against foreign invaders. That's the real purpose of these restriction enzymes. So restriction enzymes, it's important for you to know, restriction enzymes are produced by bacteria and also bacteria to defend themselves, okay? We harvest these restriction enzymes. So restriction enzymes have really funny names because the names are based off of the organism that we harvest the enzyme from, okay? Some come from Pseudomonas, some come from E. coli, etc. So there's two main categories of restriction enzymes. I'll take this point to remind you that if it's in red, it's probably important. You should know it, okay? So these restriction enzymes can cut to leave what's called sticky ends or overhangs, or they can cut to leave blunt ends, okay? So here's the author trying to demonstrate this, okay? Here's one type of, here's a restriction enzyme that it is able to recognize this restriction site. So this is DNA, okay? We're dealing with DNA. And one of the things that you need to know about these restriction enzymes that leave sticky ends is that the recognition site in DNA is a palindrome, okay? What is a palindrome? A palindrome is a word that reads the same forwards and backwards. So if you look at here at the top, it goes G-A-A-T-T-C. But if you look at the bottom in the opposite direction, you go G-A-A-T-T-C. Huh, same forwards and backwards. That's a palindrome. So a palindrome is a typical recognition site for a restriction enzyme. And then what it does is it typically cuts between two nucleotides. It always cuts between the same two nucleotides. And you can see that it leaves an overhang on both sides. And we call them sticky because this T wants to make a 2 hydrogen bonds with an A. This T wants to make 2 hydrogen bonds with an A. This A wants to make 2 hydrogen bonds with a T. In other words, these overhangs are complementary to each other, specifically to each other. So if you cut two different pieces of DNA with the same restriction enzyme, they're going to have complementary sticky ends, And that's what we want, because these things find each other in a test tube. They find each other and they stick to each other due to this complementary hydrogen bonding. Okay, so this is what the sticky ends look like. See, so now the author said, imagine that one is a piece of plasmid and the other one is a gene of interest, but we're going to cut both of them with the same restriction enzyme. So we end up with the exact same overhang. And so because they're complementary to each other, they'll find each other and boop, they stick together. Okay, very neat stuff. Okay, now we're going to go into what's called, whoops, let's try it again, the blunt ends. Okay, so if we look over here, here's our blunt end. Okay, and notice that it cuts right down the middle and there's no sticky overhang. You might think to yourself, wow, how is that going to work? And I'm going to tell you, it doesn't work well. It doesn't work well because there's no complementary hydrogen bonding to bring these two together. However, if you're willing to play the odds and hope that two absolutely different pieces of DNA come together no matter what, this is a way to do it. You use blunt ends that do not have a sticky overhang, and hopefully they'll come together. But if they do, it's not going to matter where one came from and the other came from, okay? Once they're together, use the enzyme ligase to make that last phosphodiester bond here and here, and now you've incorporated your gene of interest to your plasmid DNA, okay? So that's, this is the blunt ends. All right. The next two on the agenda are vectors. Okay, a vector is anything that can deliver a payload of some sort to a host. In this case, the payload that we're talking about is a gene of some sort. Okay, so for us, we've already discussed two types of vectors. Okay, one of them, of course, is plasmids. Okay, why? Because it delivers a gene into E. coli, and we've also talked about small interference RNA, which is a vector that can be hypothetically delivered to a cell in order to interrupt the expression of some protein, okay? So again, vectors is something that delivers a genetic payload into the host's chromosome, okay? Some of the traits that we're looking for is that they're easy enough for us to engineer in the lab. Okay, we're trying to make them so that they survive inside the cells. Again, this is a very important point because our cells are not very happy with DNA in the outside of the nucleus. Okay, evolutionarily, if DNA has been found in the cytosol, typically what that means is that that DNA or RNA belongs to a virus or a bacteria and it's up to no good. So consequently, our cells have a very low tolerance for foreign or even their own genetic material in the cytosol. This is the reason that messenger RNA gets broken up in such a short order, because our cells do not want genetic material in the cytosol. So one of the main problems in delivering genetic material as a possible medication is to have it survive inside the cell. It is not easy to do, okay? So survive inside the cell. We might want it to contain a genetic marker. What does that mean? It has a genetic marker so that we can use a complementary probe and find out if the sequence was assimilated by the host, okay? And finally, we want it to be incorporated into the genome in such a way that it actually does its job, which is to express the protein, okay? Examples of vectors, viral vectors, okay? Transposons, these are small pieces of DNA that have the ability to incorporate themselves into the host's genome. Plasmids, we've talked about those. And again, these are all delivery methods for delivering specifically genes into our host, okay? And again, these are essential to gene therapy. And again, gene therapy is the ability for us to deliver some gene into a host in order to produce a protein for that host, okay? So those are vectors, delivery of DNA, of the nucleic material, DNA, or even RNA. Here's our author's attempt at elucidating for you the manufacture of a recombinant vector. We've actually already talked about this. So here we have a plasmid. We have a plasmid, okay? And we have DNA. That DNA, forgive me, I just lied to you. And we have messenger RNA, and we use RNA transcriptase of that post-transcriptionally modified messenger RNA to make the complementary DNA here. Then that complementary DNA and the plasmid are both restricted with the same restriction enzyme. This guarantees that they both have the same sticky ends, okay? These two sticky ends, because they're complementary to each other, the gene of interest will attach itself to the plasmid, and then we use ligase in order to form that last phosphodiester bond here, and here, and here, and there. So now the gene of interest is actually part of the plasmid, and then we carry out transformation, which is to put that plasmid into the E. coli, and then we grow our E. coli, okay? So there is our form of manufacturing a protein of interest. Gene libraries, okay? Gene libraries, same concept. All we're trying to do here is take a gene. You got to understand, genes in humans are very, very long. And you're not going to pick one gene from a human and believe that you're going to stick it into a single bacteria. It's just not going to work. It's too big. So what you have to do is you have to take that gene and you restrict it with restriction enzymes, okay? And then each one of those pieces of the gene goes into a different plasmid, and then each plasmid goes into a different bacteria, and then you save all those 10, 15, 20 bacteria, and between those 15 to 20 bacteria, they hold that one gene. Then later on, you can study part of that gene by taking that bacteria and expressing that particular part of the gene, okay? These are called gene libraries. And they can be 20, 30, 40 different bacteria holding this one gene, okay? So that's what they're doing. A picture's worth 1,000 words, so here it is. Okay, we have a genome. Here are authors taking it out of a bacteria. That's kind of cute. Typically, we're getting it out of a eukaryote of some sort. We isolate the gene of interest. We're going to cut it with restriction enzymes. There it is. It costs 11 fragments. Those 11 fragments get incorporated into 11 different plasmids. And then those 11 different plasmids get introduced into 11 different bacteria. And then those 11 different bacteria get put into 11 different test tubes or lyophilized test tubes. And now if you want to study part one, you use tube one, or if you want to, whatever, you get the idea. But that's a gene library, okay? Now we're moving to something that's actually interesting. This is all interesting, but this is really fascinating, okay? And this is polymerase chain reaction. So two things to remember. One is that PCR, the whole purpose is to amplify DNA, okay? So we're doing PCR is DNA amplification in vitro. Vitro means glass or in the test tube, versus replication in vivo, okay? So replication in vivo and PCR in vitro are analogs of each other. They're both doing the same thing. They're both taking DNA and they're amplifying it, okay? So keep that in mind. What we're doing is we're trying to take DNA and we're trying to increase the amount of it, okay? These are the main steps. I'm not gonna take time to go into each one. The picture tells you more. But it's important for you to know that this is happening in a machine called a thermocycler, okay? And the reason for that is because all these steps of PCR take place at different temperatures. And so this thermocycler can be programmed to carry out a certain temperature for a certain amount of time for a certain number of cycles. And you and I don't have to do it. The thermocycler does it, okay? So that's a beautiful thing. So let's take a look at what we're trying to do here. Okay, here's a big picture. We're going to start with, not surprisingly, DNA. Okay. Now remember, we're doing in vitro what the cell does in vivo for DNA replication. So what's the first step? The first step is that we need to break the hydrogen bonds between complementary DNA strands. If you remember in replication, the guy that was responsible for this was helicase. Here, we're not going to use the enzyme helicase. What we're going to do is we're going to heat up the DNA to 94 degrees, and when that happens, the hydrogen bonds between the complementary strands break, and you end up with two strands of DNA. That event, where you heat the DNA in order to break the complementary hydrogen bonds, that's called denaturation. Denaturation, okay? Now, if you remember, in vivo, the next thing that happens is that an enzyme called primase comes in and makes a little RNA primer. And the reason for this is because DNA polymerase cannot attach and synthesize DNA unless there's a 3' end to which it can attach. So again, in vivo, we have primase come in and put in an RNA primer, okay? That is not the case in PCR. In PCR, we already have formed primers, okay? And I think our author made a mistake here. This is supposed to be an RNA primer, not a DNA primer, RNA primer, okay? Depending on how many locations you want to amplify, you need 2 primers for each location. You need one that is complementary to this side and one that is complementary to this side. They're called forward and reverse primers. Otherwise, you're not going to be able to synthesize both strands of DNA. So each loci, each location that you're amplifying needs 2 RNA primers. If you're amplifying 2 parts of the DNA, you're going to need 4. If you're amplifying 8 parts of the DNA, you're going to need 2 times 8 or 16 primers. This makes the chemistry very complicated. Okay, so we've denatured the DNA. Okay. Now we're going to cool it down a little bit to allow these RNA primers to associate forward and reverse, okay? And that is step, our author calls it priming. I don't like that name. Most journals call that annealing, okay? And that's because the primer anneals or attaches to the DNA strand. So that step where the RNA primer attaches to the strand, our author calls it priming. Most other people in the free world call that annealing. Okay, once that happens, once the RNA primer has annealed, then we heat it up and DNA polymerase attaches, okay, DNA pol 3, and that synthesizes the complementary strand of DNA, okay? That is extension. So there you have it. Denaturation, annealing, and extension. That's one step. What has happened in one step? Well, we started with 1 strand of DNA. After 1 step, we have 2 strands of DNA. If we do it again, we're going to have 4. If we do it again, we're going to have 8, then 16, then 32, then 64. You get the idea, okay? So every time that you run the cycle, you double the amount of DNA. Most cycles are around 30 or 32 cycles. By that time, you end up with millions of strands of DNA. So what this means is that if you had a couple strands of DNA at first, after 32 cycles, you have enough DNA to carry out whatever investigation you deem appropriate. Beautiful stuff, okay? And I'm going to stop here and do another recording because I don't want these things to get too long. So I'll see you guys in just a little bit, okay?